Hello everyone, and welcome to today's webinar in the Startup and ESTCP webinar series. My name is Rula D. I'm a senior principal at Geosyntac Consultants in California and the coordinator of the webinar series on behalf of Startup and ESTCP. I'll be facilitating today's call. The webinar will consist of a brief overview of the Startup and ESTCP programs by Dr. Curt Preston. After Curt's opening remarks, we will transition to the technical portion of the webinar. Today's event focuses on Department of Defense research efforts on the management of novel Hawaiian ecosystems. First, Dr. Susan Cordell of the United States Department of Agriculture will present the use of quantitative approaches towards restoration in dryland ecosystems in Hawaii. Her presentation will be followed by a short Q&A session. Next, Dr. Rebecca Ostertag of the University of Hawaii at Hilo will describe how to integrate plant functional traits into restoration design using an example from the Hawaiian lowland wet forest. We will conclude the webinar with a second Q&A session. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions well in advance of the Q&A sessions. On this slide, we have provided a few suggestions in case you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into this conference line. However, if you continue to have problems, please submit a comment using the chat box in the lower left corner of your screen. With that, I would like to introduce Dr. Curt Preston, who is the Startup Program Manager for Resource Conservation and Resiliency. Dr. Preston has tracked his career between civilian university and military positions. As the Associate Vice Chancellor for Research at the University of Nebraska-Lincoln, he led faculty development efforts to improve research competitiveness and worked with technology transfer personnel, academic departments, and colleges to build university research capabilities. He has served as a member of Chief of the Army Corps of Engineers Environmental Advisory Board. Curt?
Thank you, Rula. I'm happy to welcome everyone to today's CERTIP and ESTCP webinar. CERTIP is the Strategic Environmental Research and Development Program established in 1991 by Congress as a unique partnership between the Department of Defense, the Department of Energy, and the Environmental Protection Agency. CERTIP's mission is to identify and address high-priority environmental science and technology opportunities that focus on Department of Defense requirements. CERTIP funds both fundamental research as well as advanced technology development that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under CERTIP and other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. CERTIP and ESTCP are complementary programs, with much of CERTIP research occurring in the lab and at pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, though occasionally supporting lab efforts are conducted. There are 4 program areas in CERDP and 5 in ESTCP. The Energy and Water program area is only found in ESTCP, while the other 4, environmental restoration, munitions response, resource conservation and resiliency, and weapon systems and platforms, are CERDP and ESTC programs managed jointly by a designated program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Resource Conservation and Resiliency Program area, for which I'm responsible. Resource conservation and resiliency has essentially 3 main areas of research: natural resources, climate change, and air quality. Our webinar series highlights research and demonstration efforts from each of the 5 program areas. This is our last webinar in December, and we will resume the series in 2018 with a webinar roughly every 2 weeks. You can find more information about upcoming webinars at this link. I hope you enjoy the webinar and have a Happy New Year.
Thank you, Curt. It is now my pleasure to introduce our first speaker, Dr. Susan Cordell, who is a research ecologist with the Institute of Pacific Islands Forestry, a research branch of the USDA Forest Service located in Hilo, Hawaii. Susan's current areas of research include ecophysiology, restoration ecology, functional ecology, tropical dry ecosystems, tropical ecology, grass invasions, and Hawaiian ecosystems. For most of her professional career, Dr. Cordell has focused on the conservation, preservation, and restoration of tropical systems. Susan received her Bachelor of Arts degree in aquatic biology from the University of California, Santa Barbara, and a Master of Science and doctoral degrees in botany from the University of Hawaii at Manoa. Susan?
Hi, good morning everyone, or at least it's morning here in Hawaii, and aloha. I'm really happy to be here and to be presenting this work to all of you today. I'm going to start off the presentation with the idea that there's really no recipe for plant restoration in degraded ecosystems, and in particular when you're making decisions about what and where to plant. And in fact, even the idea of baselines and what we restore to is really changing as everything is becoming a moving target in today's world. And this uncertainty often leads to marginal success of projects, as well as utilization of excessive resources. So my goals for the presentation today are to convey to you the challenges that we face here in Hawaii and the Pacific Islands regarding restoration, and then hopefully convince you why this is an important issue for biodiversity on Department of Defense lands. And then I will elucidate some strategies that we have come up with for restoration approaches and planning. So I'm going to lead you through this process where we really developed and to tackle the question of actually where to plant. And Becky later in the presentation will feature another process that we're working on regarding what to plant. So in this presentation I'll be using a case study of a military training area in a tropical dry forest in Hawaii. So just to give you some background for those of you not very familiar with our ecosystems here, tropical dry forests, and not just in Hawaii but around the world, are declining at alarming rates. And in the case of Hawaii, this is primarily a result of fire, which originates with the invasion of native ecosystems by fire-prone exotic grasses. And in Hawaii, many of the Department of Defense installations occur in these tropical dry forests because they're very suitable for training. But also these areas contain a very large proportion of the threatened and endangered species. I think more than 25% of the plant species listed in Hawaii occur in these ecosystems. if you look at Department of Defense budgets, they spend more than $10 million a year protecting these threatened and endangered species, and often resulting in compromised ability to train. So if you look at focused restoration planning, this will allow more efficient conservation of biodiversity, reduced management costs, and effective partitioning of multi-use landscapes. In Hawaii, the land base is very compressed, so many of the landscapes have multiple uses. And in the case of Department of Defense lands, and where I've been working, there's also recreation activities in the form of hunting that's occurring on these landscapes. Okay, for some more background. What I have here is just a depiction of the grass fire cycle that many of you are probably familiar with. Essentially, you start off with an intact native forest, and you have various pressures on the landscape, land use, ungulates, grass invasion, and then you get a grass-invaded forest. I have a picture there on the right where you can see sort of what that looks like. And then eventually another fire comes in and burns the remaining overstory canopy trees. And because they're not very resilient to coming back after post-fire, what you get are these monotypic grasslands coming in, and then that just perpetuates this grass fire cycle. So our original question with our original CERDIP funding was, how can we develop tools to break this grass fire cycle? Because we do know that native dry forest species prefer shade and protection, and we also know that invasive C4 grasses need full sunlight. So our idea was, can we reduce grasses by planting trees and shrubs to reduce the grass layer? So our first question in starting to do this was, well, where do we do this first? And we have a lot of things to contend with here, as any restoration projects do, and I'll just go over some of those. So some of our main challenges to recovery in this particular system is that survival is low and variable. So if you look at this figure here, it's number of outplants on the y-axis and the year planted on the x-axis, with the clear bars being the total outplanted, and then the hatched bars being the ones that survived. And you can see that it's highly variable over time, but in general about 30% survival over time. We also have very extreme environmental conditions in many ecosystems in Hawaii, and Pohakuloa training area, which is the area that I've been working in, is no exception. Rainfall is not only low, but highly variable inter and intra-annually. And so you can see on this figure, this is different years from 2004 to 2010, precipitation on the y-axis. And you can see not only is there huge variation over the years, but you can see that the different colored bars are the different months of the year, that there's not a consistent pattern of when we actually receive rainfall. And if you look at 2010, you see sometimes we have these very catastrophic years that make it virtually impossible for plants to survive. Unfortunately, this is when we were actually carrying out one of our CERDIP experiments, so we had to do some modifications for that particular project. Let's see. Other challenges that we face, I think one of the biggest stressors to plants in this particular environment is desiccation stress. It's very, very windy in this ecosystem, and plants really, really suffer during these dry periods and windy periods from desiccation. So I probably should have had this slide earlier to show you where I'm talking about, but a big challenge to recovery on Hawaii Island is large areas with limited access. So here's a slide of the Big Island of Hawaii, and you can see I have Pohakuloa training area outlined there. And it's between two large mountains, Mauna Kea and Mauna Loa, and it's a 40,000 hectare landscape with 15 threatened and endangered plant species. So it's very challenging for land managers to figure out where to do things and how to get there. Okay, because Pohakuloa training area is so large and must serve multiple objectives, we chose to take a landscape approach using remote sensing tools, and two, to be able to have some kind of indicator to be able to quantify our restoration success. So this is how we started our eyes in the sky and plants in the ground approach to restoration and reintroduction. And I've had the pleasure of collaborating with Greg Asner, who developed and maintains the Carnegie Airborne Observatory. And together we put in a collaborative proposal to combine remote sensing tools with field-based approaches to strategically look at a landscape and its potential. So when we've decided upon our tool, some of our first questions were, what are we looking for with that tool? And where on the landscape will outplanting be the most successful? And if you look at some of these arrows, would you start under an existing canopy for protection from stressors, or would you plant in a protected area at the base of a hill, or would you plant on the surface area? So we started thinking about this and walking around a lot on the ground doing ground truthing, and started sort of thinking, like, if I were a plant, where would I do best? And really looking around at how the vegetation is responding to different areas. So that's really sort of how we started. We had the tool, got the information, and then sort of tried to figure out how to use it. So our goal was to fly the plane, and then thousands of hours of processing time, as always these things take, and produce a high-resolution digital elevation model, thinking that microtopography is likely very, very important, with a 2.2 meter pixel scale. And this is of the entire area of Pohakaloa training area. And from this we would produce a map of some kind of suitability, and I'll get into that next. Okay, as I mentioned previously, desiccating winds are a huge stress for plants in this area. So our first criteria was to develop a habitat suitability map to determine the prevailing winds in this particular area, such that areas that are topographically protected from the wind get a value of 1. And you can see the low suitability pin there on the landscape. And when you're not in a protected area, you get a 0. So you see that high suitability and low suitability pins in there is where these would occur on the landscape. The second criteria was to figure out where on the landscape resources would be most abundant. And topographically, this would be areas that have descending topography, because this is where nutrients and water are most likely to accumulate. So using this criteria, a value of 1 would be given to areas that are sort of at the elbow of a crater. You can see the high suitability area there. So not necessarily at the bottom, but sort of at the elbow where things are accumulating. And so if you were in that area, you would be given a 1, and areas that would be given a 0 would be topographically flat or too steep, so at the very bottom or at the top of a ridge. Okay, so when you put it all together, 1, 2, or 0 criteria met, you have a 2.2 meter pixel map of habitat suitability for a 40,000 hectare landscape. So you can see here, this is the entire area of Pohakaloa training area. You can see the sort of breakout of a half-kilometer area and how diverse this landscape is in terms of microtopography and suitability. So you can see on the photo there, the orange arrow represents the low suitability area, and the blue arrow represents the high suitability area. And if you look at the entire landscape and break this down, you see that for the most part, the bulk of the landscape at Pohakaloa training area is actually in the 0 or 1 category, with only 15% in the 2 category. Okay, so this is all great as an exercise in the lab, but how about what is actually happening on the ground? So to test this, we set up an experiment across the landscape where we selected high and low suitable areas and measured biotic and abiotic variables to determine the viability of the model. So here you can see that wind speeds are an order of magnitude higher in the low suitability areas. So it looks like we got that feature right. So here is average daily wind speed on the y-axis, and high suitability as the dark symbols and low suitability as the light symbols. And again, you can look in the photo and see the pictures of our weather stations in these high and low suitability areas. we also measured existing plants that occurred in these areas across the landscape in our plots to look at evidence of topographic suitability. And what we found is that plants are taller in the high suitability area. They have higher levels of foliar nitrogen in the high suitability area, and the leaves are wetter in the high suitability for most of the year. And we measured this with these leaf wetness sensors. And also, to our delight, the Pohakaloa Training Area environmental staff had spatially explicit locations for every single endangered plant species across the landscape. So this provided us a perfect opportunity to look for potential habitat associations. So we overlaid the GIS of all of the plant locations with our habitat suitability model to look for these associations. And here's a picture of Haplostachys haplostachyi. This is one of our mintless mints in Hawaii. It's a beautiful plant with very fuzzy leaves and beautiful white flowers. And you can see the green points on the map show the populations of this particular species. And so what we found out is there were some strong habitat suitability associations with these existing threatened and endangered species. 6 species had higher suitability, 2 species were associated with lower suitability, and 3 species had no association. So our focus was really thinking about these 6 species that were associated with high suitability. The 2 species with lower suitability is also of interest, and I have some ideas for thinking about how we might actually utilize that information, but that might be another webinar down the line. Okay, then as a final test, we planted native species across this experiment. And as you can see, survival was higher across the board in the high suitability areas. So it's a sampling data on the x-axis and survival on the y-axis here. So we feel really good about this tool. I hope I've given you a convincing argument that these tools play a valuable role in helping us to determine where to plant in these tropical areas. I want to share with you all an extension in ArcMap that was developed by one of the colleagues on our project, Aaron Questad at Cal Poly Pomona. And if you use this extension in ArcMap, you can create your own habitat suitability model for dry areas. So this is, again, using the microtopography approach. So users will need to access to a high-resolution digital elevation model, and I would say less than 5 meters resolution would be ideal, and an understanding of prevailing wind direction in your area. And most times winds tend to be variable on a diel pattern in areas, so I think it's best to select the direction of wind during the photosynthetically active part of the day, because I think that's when it's most meaningful. Okay, so taking this information, we wanted to sort of address some other tools for helping not only select where to plant on the landscape, but where do you actually, if you're doing restoration planning, how do you look at a landscape and break the landscape up into what is the most suitable area for different activities? So using the Carnegie Airborne Observatory plane with the LiDAR, we added hyperspectral imaging measurements to this. And by using this technique, you can break the landscape into differing vegetation patterns. So in our case study, it's a pretty simple ecosystem. So you have just several types of ecosystems to define. For example, we have grass-only ecosystems, lots of areas with just bare ground or rock with very little vegetation, areas that have trees and shrubs with an invasive grass understory, and then other areas that are actually, for the most part, native trees and shrubs without the grass. So there's a lot on this figure, so I'm going to take a little time to break it down for you. And first of all, I wanted to tell you that this area here represents about 10,000 hectare piece of land at Pohakaloa Training Area. And I'm going to use the arrow here. So as you can see, these gray areas here represent areas with little or no vegetation and are mostly flat. The pink areas So you can't see it so much on the big map, but you can see it down here on the inset. These pink areas represent areas with invasive grass understory with a tree shrub overstory. These yellow areas here represent trees and shrubs without a grass understory, and then these green areas here represent monodominant stands of invasive grasses. So if you look at the entire landscape and you're trying to figure out where to do what activities across the area, if you are interested in restoration to reduce fuels or to expand your areas of conservation, you would select areas that have high fire risk, so that would be high grass understory, but high suitability for plant growth. So that would be the areas that have a tree and shrub overstory. So those would be the pink areas. If you're interested in restoration of endangered plant populations, so your very high biovalue plants, you would target the areas with low fire risk and high suitability for plant growth, and those would be the yellow areas. And for portions of the landscape where you're really focused on physical fire barriers or protecting high-value conservation areas, you'd want to target areas with high fire risk and low suitability for plant growth. And in particular, for those particular areas, it would be best to focus those fire management efforts in areas like this where you're protecting high-value biovalue areas. So you can see across the landscape here that there's really large monodominant stands of grasses up in here that have that you might want to focus your efforts on fire control. And then also if you look at this particular area here that's all gray, that might be a really suitable area for training because you don't see a lot of high biovalues interspersed in there. So I think it really gives you a good overview of the landscape for protecting these areas. And we just have a paper out in Restoration Ecology if you would like to learn more about this. Okay, so that brings me to my last slide. Oh, I got to get rid of these here. Sorry. Okay. And I guess in conclusion, we've been very happy with the Department of Defense supported research program that has served as an important catalyst for generating this really exciting science to help address really critical natural resource management needs in Hawaii and the Pacific. And this habitat suitability model allows users to identify areas of the landscape where stakeholder activities can be prioritized based on biophysical and geomorphic characteristics. And this management strategy enables the installation to conduct training while conserving natural resources upon which the quality of training ultimately depends. So I thank you very much, and I think I have some time for questions.
Yes, thank you so much, Susan. We do have time for questions. And I'm going to go ahead and start with this one. Can you please share with us what folks can do if they do not have access to high-resolution imagery?
Yeah, that's been a question that we get asked a lot because there's a lot of people that have been interested in this particular approach. And I think what we're starting to learn is there's actually a lot of imagery out there that people aren't aware about. So I think working with remote sensing folks that have an understanding of different resources that are available would be a first step to do. We're also working with satellite imagery. Satellite imagery is getting a lot more robust in terms of resolution. So and we did find that there is a strong correlation with the use of LiDAR and satellite imagery for creating high-resolution digital elevation models. And I think partnering with sort of multi-agency approaches to look at landscapes so that if there is a bill of acquiring this information, that the resources can be shared.
Thank you, Susan. you mentioned that some plants have associations with low suitability habitats. How do you factor this into your approach?
Yeah, it's interesting because you can't just say, well, we're only going to care about these plants that occur in the high suitability areas. And we do have to think about associations with the lower suitability areas and conserving those particular landscapes as well. So I think that's really trying to get an understanding of the plant physiological strategies associated with adaptation to these stressful environments. And so I think what that's going to really take is a much more specific approach to studying individual plant populations and their requirements.
Thank you. And can you tell us how your technology may be used, if possible, in other ecosystem types?
Yeah, we get this question a lot because certainly tropical dry forests aren't the only ecosystem that you're going to want to work doing restoration. And we've started thinking about using light, using remotely sensed light information for tropical wet forests here in Hawaii, and helping looking at quality of light and potential gaps in the area which are more suitable for restoration. So I think that's one of the approaches we can use, and I think really the sky's the limit. Depending on the stressors of your ecosystem, most likely you will be able to get some relevant information using remotely sensed techniques.
Wonderful. A question from the U.S. Forest Service. Are endangered plants being planted in some of the areas you identified? And if not, are there plans to do so?
Yeah, that's a great question. We've worked really closely with Pohakaloa Training Area environmental staff, and we have conducted a project with funding from ESTCP to actually plant endangered plant species in these high and low suitable areas. And the Pohakaloa Training Area environmental staff has been very interested in this approach, and they have access to all of the habitat suitability maps and are using those for not only monitoring and planting endangered plant species, but looking for additional plant species that might be on the landscape. So there's several uses for this, and not only is it working with Department of Defense managers, but also all land managers in Hawaii that have been shown this technique have been really excited about it.
Thank you. How might more pronounced extremes in environmental conditions affect your approach?
Well, I think that I think more extreme conditions that are expected or predicted for tropical dry systems in Hawaii are for them to become drier, more arid over time. So my guess is that the habitat suitability would be even more critical during those times, because if you're selecting areas on the landscape where the microtopography is suitable for resource accumulation and away from desiccating winds, I think that it should only strengthen the usability of this model.
Excellent. And just to wrap up this portion of the webinar, what are the next steps for your work? What's next?
Well, I think that there's other components of this that we're really interested in exploring from a scientific basis. I'm very interested in wind deposition and how habitat suitability is playing a role in that, because that might be related to some of the increases that we're seeing in nutrients in these particular high suitability areas, as well as perhaps loss in the low suitability areas. I also think from the term of application, we're really trying to get users connected with high resolution imagery. That seems to be the biggest need right now, is to have a connection with people being able to access this imagery. So I would say that's our biggest challenge and our biggest desire to overcome right now.
Thank you so much for a great presentation. I would like to remind our audience that you can continue to submit questions in the chat box on the left-hand portion of the screen, and we'll be able to revisit your questions in the final Q&A. The next part of the webinar will be presented by Dr. Rebecca Ostertag, who is a professor at the University of Hawaii at Hilo. Becky is a forest ecologist who studies the functioning of tropical ecosystems. This focus has led her into the field of ecological restoration, in which she uses ecological theory related to functional traits to design restoration treatments. Since 2002, she has been working in the lowland wet forest ecosystem at the Kiukaha Military Reservation. She has served as a PI on several startup projects to develop novel techniques for species selection. Becky earned a bachelor's degree in biology from Carleton College in Northfield, Minnesota, and a doctoral degree in botany from the University of Florida in Gainesville. Becky?
Yes, I'm very happy to share our research with you today, and I want to go through an agenda of what I'm going to be discussing. I'm going to first talk through concepts and applications related to plant functional trait theory and measurements in restoration. I'll define what functional traits are and explain their utility specific to restoration ecology and management. And then I'll provide an example using our startup-funded hybrid restoration project in Hawaii, which we call the Liko Napalina project. That example is intimately linked with a computer tool that we developed during this startup project called the Restoring Ecosystem Services tool. And I'll explain that tool through the example, and then I'll end with some suggestions for future applications and how I see that this approach can be applied beyond our own Hawaiian ecosystem. So there's many challenges in restoration, and anyone that's familiar with the subject knows these challenges well. Restoration is often a series of best guesses, and our research is trying to see if we can take those best guesses to a more planned, strategic set of conditions. First, when doing restoration, it's often very subjective because we have limited information. We may not know the original species composition, the life history characteristics of the species, and there may be many other biological unknowns. Second, in many cases, we lack an appropriate reference system or a historical baseline. And so in those cases, we cannot do true restoration, getting back to an original historic state, but we must aim for something else, something like rehabilitation, which may be getting to some stage but not complete restoration, or potentially a replacement with other completely new species. Even if we could get back to a previous species composition, our planet is no longer the same because we've undergone climate change and other factors, and so really getting back to the original conditions is not always realistic. And then we need to consider future climate change, species migrations that may happen in the future, changes in the disturbance regime such as drought and fire, and all of those are important considerations in restoration planning. And finally, from a practical standpoint, every restoration plan must take into account labor and time and cost. These are tremendous logistical issues that need to be balanced with the considerations and demands of stakeholders. So planning for restoration is not a simple feat. It's a very complicated dance, and the goal here is to start to do that dance in a way that's not completely haphazard. One innovation that's gaining traction is the use of functional traits in restoration. Plant functional traits influence growth, reproduction, and survival. They are characteristics that we can measure that are proxies, and they reflect resource use and life history trade-offs. As an example here in this slide, you see a pile of leaves that we've collected from study sites in East Hawaii Island. And you can see that these leaves vary in many ways. They vary in leaf shape, in leaf size, in petiole length, in their color, in their presence of lobes or teeth, in their thickness. And all of these things are traits that could influence the rate of photosynthesis and the rate of water loss through the stomates. One convenient thing about functional traits is that they vary consistently among species, and there's often convergence in trait values within a given environment. For example, we might expect in dry environments that plants will have thicker leaves. And we can therefore use these traits to understand plant adaptations. And that's because functional traits do relate to ecological processes. In this top figure, what I'm showing here is a gradient in leaf characteristics representing the slow to fast continuum. And the reference on the last slide by Peter Reich, and on the bottom here, this Reich 2014, is a really good starting point for reading about functional traits. He calls that his manifesto, and it's very, very comprehensive. Going back to the top panel here, this depicts the slow to fast continuum that Reich talks about. On the left side, we see that we have leaves on the slow end of the spectrum. These leaves have low rates of photosynthesis, they have low foliar nutrient levels, but they're often built to be durable and have a long lifespan. At the other end of the spectrum, leaves on the fast end of the spectrum tend to be use resources more intensively. They tend to obtain higher nutrient levels and photosynthesize at a faster rate. But there's a cost, and that cost is being thinner and less durable and having a shorter lifespan. At the bottom end panel here, there's two graphs that show wood density. One of them in relation to relative growth rate, and the other in relation to mortality. And you can see that as wood density increases, trees grow more slowly and they have a lower annual mortality rate. So a high wood density represents the slow end of the spectrum, whereas a lower wood density correlates with faster growth rates but a higher mortality rate as a trade-off, and that correlates with the high end of the spectrum, the fast plants with much more fast processes and on the fast end of the slow to fast continuum. This is a slide that shows how we can use restoration traits, functional traits in restoration, and it really depends on the restoration goal. I've listed some examples here. So if we're interested in creating ecosystems that will be able to sustain wildfires that might increase in the future due to climate change, we might select species that have thick bark, a high water content, an ability to resprout, and have seeds that could survive fire or perhaps that have fire-stimulated germination. We can collect these data on traits from a variety of candidate species and then select those species with the most appropriate trait values. Similarly, you can see listed other restoration goals, drought tolerance, carbon storage, and successional facilitation to be able to move a system from one set of species to another set of species. And these traits are all related to these different goals, and depending on your goal, you may choose different traits to measure. When I explain the computer tool in a few more slides, these 4 restoration goals are listed in the computer tool and have these suggested traits associated with them. So functional trait-based restoration can be flexible. Okay? It doesn't necessarily need to return to a previous state, but it can allow for species substitutions that can be made with the goal of restoring important ecosystem functions and services. The basic premise of functional trait restoration is to use functional trait values to make species decisions and develop teams or groups of species that will work together to meet the restoration goals. So this is a quick how-to guide that I've put together to sort of simplify what I'm talking about for a general user. This is the approach that you would take. The first step is to define the goals, and this is a process that should involve scientists, managers, and other stakeholders. Then you would choose traits based on your restoration goals, such as what I showed you in the last slide, and also take into consideration the ecosystem in which you're working, which is going to influence your goals and your choice of traits. Now, trait data are complicated, but they can be compiled. In many cases, data already exist in global databases, or they may have collected in the literature, or you may be able to find that information from researchers working in your local area or at a particular site. If you don't have any trait information, you can develop a plan to collect trait data. And some traits like leaf area are very easy to collect and almost anyone can do it. Other traits like photosynthesis rate are more difficult because they require equipment and some expertise. These can then lead to the species decisions. We advocate using a statistical method, some kind of ordination. In our case, we're demonstrating principal components analysis, or PCA. And this ordination technique allows you to see the spatial relationships among species, how similar they are in their trait profile, in their trait values, in a multivariate dimension. And seeing these spatial relationships among species can help you to make species decisions about which ones to include in your restoration and which ones perhaps to not include. This is a rare forest type where we're working that I'm going to be explaining our case study. It's part of our startup-funded project, and it happened at Keaukaha Military Reservation. It's lowland wet forest, and lowland wet forest is a unique type because it only exists now in remnant patches. It was much more abundant across the Hawaiian Islands, but it was cleared for people, for agriculture and other development. And the clearing began first when the early Hawaiians came, and then later when further Westerners came, there was larger scale clearing for things like sugarcane. The site at Keaukaha Military Reservation is on an aa flow. That's a chunky type of lava that's difficult to walk in and maneuver around. And that's probably what saved this site from agriculture, because it is very low elevation, only 30 meters, and that is in the zone where many people lived and conducted agriculture. It's a wet site. It gets about 3,400 millimeters of rainfall a year. The canopy still has some native species, and that's what our interest is in the site. We're not starting from ground zero. We're starting with a site that has native species and that our previous research has showed is producing seeds that could be important propagules to be used in restoration. However, the site is heavily invaded. The understory is almost completely non-native, and most of those non-native plants score very highly in the Hawaii Weed Risk Assessment for their invasiveness. So they've been demonstrated to be highly invasive. And many of these species that are trees are also getting into the canopy. So they're crowding out the native canopy, and they're inhibiting regeneration of the native understory. We initiated the Liko Napolina experiment that I'm going to talk about in 2014, but the work that we've been doing to lead up to this was several years of planning that went into designing this restoration experiment. And in Hawaii, we think that restoration is taking this approach because we have such a tremendous threat from invasive species. It's our biggest challenge in restoration here. Approximately half of the flora in Hawaii is non-native. And the unfortunate reality is that in an increasing number of ecosystems, it's really not feasible to return to a previous state. There's reasons include a lack of a reference site or historic baseline conditions. This certainly applies to our study site at Keaukaha Military Reservation, these lowland wet forest sites. We do not have good pollen records or good records of species composition before humans got here, and we don't have any sites that are uninvaded. So every site has been altered by invasive species, and not just plant species, also animal species. Feral pigs are a major problem in the lowland wet forest, and they've been invaded by ants and mosquitoes and many arthropods as well that are non-native, as well as in many cases also by coqui tree frogs. And so we have a whole menagerie of new species. We've developed a novel ecosystem in these remnant lowland forest types. we named our experiment Leuconopolina, and that's the case study that I'm going to be explaining, because that means growing new relationships, or budding new relationships. And what we're doing is using these restoration goals that we've defined of invasive resistance, carbon storage, having a clear understory for military training, and native seedling regeneration, and using that in this concept of a hybrid ecosystem. The hybrid ecosystem is a mixture of native and non-native species. The non-native species are species that we've carefully chosen to be non-invasive, and that's based on our observations of visiting many areas in the lowland wet forest climate zone, and also using the Hawaii Reed Risk Assessment, which is publicly available and has data for many species, giving them an assessment of their risk. If they gain a score of 6 or greater, then they're likely to become a pest in the Hawaiian Islands. To meet these goals, we, as I mentioned, focus on this hybrid ecosystem of mixing native and non-native species. We used a large number of species in the beginning to, as candidate species, to test out how they would occur in their functional trait profile. We used 15 traits that emphasize leaf, wood, and reproduction traits. And for many of the species, we collected our own data. Some data we found in the literature. And then we put this information through our Restoring Ecosystem Services tool. What we did in the Leuconopolina experiment is we decided to take an approach where we would clear all the non-native, which were pretty much, in our case, all invasive species. And we did that by creating 20 different plots, each 30 by 30 meters. The plots had 4 of those plots were controls, or what we're calling references, and they were left as is, so they were not cleared. The other 16 plots were cleared of all non-native species, and we left native trees standing. You can see in the figures here, the one at the bottom is what the forest looked like before we started, and the one at the top is what the forest looked after we did our clearing. So what's standing are native trees, and you can see pretty much everything else has been taken out, and the understory is pretty bare. This approach may not be appropriate everywhere, but in Hawaii we have an extreme invasion problem. Our current condition has led to us having to take this kind of broad-scale approach to test it, and we're using this as a test bed. Such extreme conditions may not exist elsewhere, and so Hawaii becomes a great example. We're sort of an extreme at the end of the spectrum. So our goals are to replant this mixture, species mixtures in this restoration experiment with specific combinations of functional traits, and we use this computer program that we developed, the Restoring Ecosystem Services tool, to help us get there, and then we established long-term monitoring. That long-term monitoring is still ongoing, and we're interested in understanding how these forests will develop over time as we've created these new hybrid ecosystems. We developed a planting design that I'm also showing, and that planting design, I'm not going to emphasize this here, but the colors represent different species, and the sizes of the circles or the symbols represent the approximate size of what we expect the adult trees to become. In terms of the functional trait sampling, as I mentioned, we did 15 traits. For others to do this, some of these traits can be sampled easily and cheaply. All you might need is a way to measure leaf area, which can be done with the leaf area meter that's in the top right, or simply can be done with scanners and computer programs. Some of these traits can be found in the literature, things like seed size. Kew Garden has a database for that. There's other global databases such as TRI, which is a major database that's available. The Hawaii Reed Risk Assessment has data on individual species. And in the REST program that I'm going to be explaining, we're putting in information from global databases So some of that will be available, particularly if you have a species that's common and well studied. Here's a list of the functional traits that we sampled for the Leuconopolina experiment. This is our case study. We included a variety of leaf traits that you can see here on this slide. At the bottom, we have stem-specific gravity, which is using a small piece of stem instead of the trunk, but is directly correlated to wood density. And on the next slide, we have additional traits that we measured. These traits relate to water use, maximum height, seed mass, and variables that relate to canopy architecture. And we're really interested in canopy architecture in our case because we know at our study site that light is the primary limiting resource. Our past research demonstrated that lack of light was inhibiting native generation. However, when we open up the canopy, our past research shows that that allows for non-native weedy pioneer species that are highly invasive to come in. And so our overall goal is to build a forest with the right light environment, enough to allow native regeneration and military training, so there's a clear enough understory, but also to have invasion resistance so that it's dark enough to keep out some of the weeds, the highly invasive plant species that we don't want. So this leads me to explaining the Restoring Ecosystem Services tool. This is a computer program that we developed as part of our CERTA project. The point of REST is that it creates a visual of species relationships to each other in trait space so that you can see how species may share similar or different trait profiles. It uses principal components analysis to do this. The user doesn't necessarily need to know anything about PCA to use the program. However, if the person is familiar with PCA, detailed statistical information is available. Now in this slide for Leuconopolina, what I'm showing is an example of the use of REST. So we uploaded a file, a CSV file with our species information. We chose our restoration goals. And based on that, this principal components analysis was generated. And I want to focus on the 4 parts here in the red boxes. Each point on the graph that's numbered here is a species, and there's a parallel code that gives what each species number is, but that's not important for this example. In our case, we made some decisions based on this graph that I'm going to focus on here. In terms of right here between number 6 and number 7, both of those were ferns, and we decided that we really only needed 1 fern within the experiment, so we took number 7 out. Okay. In the case of over here with 19 and 14, 19 was a species that we knew that didn't reproduce very well, so we decided that we didn't need that species and could take it out. Number 4 over here was hard to propagate, but since it's similar to number 28, we felt like we could get rid of that species from our list. Number 13 and 29 over here, 13 was at the edge of its range and we were uncertain how well it would do, and similarly number 29 was an uncommon species that would be difficult to find propagules, and therefore we took that out as well. And then we took some other species out for other reasons, and that then led us to redo our principal components analysis. So we did it again with fewer species. Now we have the 21 species that we've actually used in our experiment. And by doing that, now we can develop further our approach. As I mentioned before, we wanted to test if functionally diverse community was better in restoration for invasion resistance and other things like growth rate. So we wanted a species with complementary species mixes. To choose complementary species, we chose species that were far apart in trait space. We want to contrast that with redundancy treatment, where species are similar in their traits. So we would choose species over here where species are similar in their trait space. Okay. And this led us to develop a final assessment of our treatments. The way that we did this is we focused on species that had slow or fast rates of carbon turnover, because carbon is turning over very quickly in our system because of the highly invasive nature of most of the species. We wanted to encourage species that had higher rates of carbon storage and slower rates of growth. So we use those 4 carbon species as an anchor in our treatments. So we have species that focus on slow carbon turnover or species that focus on moderate carbon turnover. And then we chose species using the REST model that I showed you and their distance and trait space to be either complementary, so far away from the core species, or redundant, so close in trait space to the core species. So these are the final 4 treatments with the species all laid out. Okay, now this is complicated. What if you don't know much information? The literature is suggesting that there are about 5 traits that you can focus on that seem to be the most important. So if you don't have a lot of information and you don't know what to do, these 5 traits are the ones that are recommended because they represent trade-offs between growth, survival, and reproduction. There's many benefits to this approach. We can demonstrate here that we've used ecological theory to choose species for restoration, but that theory has practical benefits as well that is advantageous to the Department of Defense and other restoration practitioners. It's using more of a trial and error approach that we think we would like to avoid. So we would like to, using this REST tool and kind of a concentrated effort in choosing species, make a very strategic plan. And REST is really meant to be a cost-effective tool. It's available free. It simplifies the process for managers, and it's iterative. You can be using it iteratively so it can allow for simulations. So in conclusion, functional trait restoration can be much broader than this example. I showed a case study that works in one type of Hawaiian ecosystem, but the strategy to use functional traits to develop a team of species and using the REST program can help you do that. If you have certain goals, those goals go in the very beginning into planning your restoration and choosing your species and developing your team. And this kind of restoration can be advantageous to reduce surprises and costs. If you have a sense of species' competitive abilities, you may be able to predict failures and steer towards species that are more likely to be successful. This has been an extremely collaborative project. Many people have worked on the project. I just wanted to emphasize them all here. And it's an experiment in process that we hope to keep going for decades. And we would like to keep measuring so that we can understand what are the short and long-term benefits of these different species mixes. So thank you very much for listening, and I'm very happy to take other questions and also contacting me separately if anyone has questions beyond this webinar.
Thank you, Becky. We've received a number of questions for you, starting off with the following. Can a functional trait-based restoration work for other goals besides invasion resistance?
Yes, definitely. I think this approach is very flexible because it all starts on what your initial goal is. So if your goal, for example, is fire tolerance because you're going to be working in a system where wildfires are likely to become more prevalent, you may just select, instead of selecting a balanced team like we did that was spread apart in trait space, you might select species that have traits that are very similar in trait values to having thick bark and high water content, for example. And so you could use REST to view a bunch of candidate species in trait space and then pick species that fall in the area of the graph where they're similar in those kinds of traits. And so it's depending on your goal, I think it's flexible. It doesn't have to be used for invasion. That's just one application of it.
Great, thank you so much. Can your approach be used for organisms besides plants?
I think it can. I was listening a few months ago to a talk about coral reef restoration, and it struck me that there's no reason why this approach couldn't be used for corals. in developing a new reef system, which they were doing in Hawaii in terms of replacing species that have been damaged or have been lost due to global temperature and bleaching events. And I don't think that this is at all necessarily specific to plants. All organisms have traits or characteristics, and so it's really just applying basic niche theory and competitive exclusion theory. By looking at species and looking at their similarities in their resource use, you can develop a new kind of ecosystem. So it is, in a sense, potentially building designer ecosystems or manipulating them in certain ways that will allow for more success in the future, because we are underneath different conditions than we faced in the past. But I don't think it's at all specific necessarily to plants. This could be done with fish species and looking at traits related to fin size. It could be related to other species related to body mass or wing morphology. I think that the possibilities are really endless in terms of that. It's just sort of a general theoretical approach that can be applied to lots of different problems.
Thank you, Becky. Another question from the U.S. Forest Service. Can you clarify whether the species you used were native, or did you use a mixture of native and non-native plants?
Yes, thank you for that question. In our example, in the Liconaupolina experiment, we used a mixture of native and non-native species. And I didn't show this graph, but a previous research had shown in our case, the native species tended to be clustered in the principal components analysis. So they were very similar for the most part in their trait characteristics. And that may be because Hawaii is long distance, most isolated island in the world, and so only a certain set of species were able to have the traits necessary for long distance dispersal to get here in the first place. Or it may be that among our native species, we've lost species through extinction, and so we may be missing some representation in the functional trait profile. I'm not really sure, and I don't think that we'll ever know the answer, but what we did see was that non-native, non-invasive species, sometimes we call them exotic species, they had in general a different trait profile. They were in a different part of the graph than the native species were. And so in our case, we're mixing native and non-native but non-invasive species because we think we will get a more functionally diverse ecosystem that way that we think will better resist invasion. And we use the Hawaii Reed risk assessment and our personal knowledge to choose species, these non-native species that we think will not be invasive. And none of the species are new to this part of Hawaii. They were all found in East Hawaii. So we're not bringing in species that are not in Hawaii or not even in this region. We're just conglomerating them together in a new place.
Thank you so much. Another question for you relates to the REST program, which you described. How can our audience members and other interested parties get a copy of the REST program? And can you tell us whether there are any planned updates to that program?
Okay, that is a fantastic question and something I forgot to mention. Right now, if you go to my website, or the Liconaupolina website, there's a link there where you can download the REST program. Be aware that it might have to go through your filters with your virus things, but it's not going to cause a virus. So we have a version of the REST program there, and we are working on writing up the user guide as a USDA Forest Service General Technical Report, and that's in its last stage of review. We are going to be doing updates to the program as we add more information and get more trait information from more species. We'll be updating it, and we'll keep it current on the website and then keep updated copies of the user guide. And if people can't find it, they can also contact me with my email on the last slide, and I'd be happy also to give them the link.
Great. Thank you very much, Becky. A couple more questions. How can this type of restoration work on a larger scale than the one you described in your demonstration?
Okay, that's a great question. The scale is very, very important because we're doing an extremely intensive experiment by removing all the non-native species and doing these plantings. And it took us almost a year to do the removals. These are large trees that we were chainsawing out of the forest, and this is done in a lowland wet forest, a tropical forest, and that is sort of like the end point, the most difficult conditions. On a larger scale and in other ecosystems with different restoration goals, I think this could be done on a much larger scale. First approach that I would do is simply to go through the process and determine what are some of the key species that are going to be doing best in your environment based on their functional traits, and then work on planting or seeding or encouraging the growth of those species, perhaps increasing their density across the landscape. And then, if it's possible, I would then start potentially doing plantings of other species that might complement the key species in the system. I feel like it can be used on a larger approach. It may not be as intensive in terms of the amount of planting, but by manipulating the environment and choosing species that will do some of the work for you, for example, controlling the light environment, those are the kinds of things you want to think about in developing this into a broader scale, is to, as much as possible, use the correct species that will change the environment in ways that will be more beneficial for the long term. And that's more what this planning process is about. It's taking a step back and saying, rather, I'm going to try this species, and this species, and throw them all in there as a gardening approach, to start being strategic about which species you choose and have reasons backed up by data to make this a larger scale approach.
Thank you so much. Now, as we try to wrap up here, I'd like to ask questions that both you, Becky, and Susan can answer. So
Sure.
Starting with Susan, perhaps, can you
Okay.
please tell us or elaborate on what lessons can be learned from Hawaii that can be applied elsewhere? Because it would seem that the problems and restoration challenges that you both described that are associated with Hawaii seem unique and extreme. Susan?
Well, I would say that I think, for those of us working here, we feel that even though while the challenges are great, we also have a system where it's somewhat like a model system where we can control a lot of factors that are really challenging to control in other places. For example, we're a tropical system, but we have low species diversity. So it's, I think, a lot easier for us to be able to test some of these theoretical ideas about systems and how they relate to restoration because of this. So we have species that occur across broad gradients. And then on top of that, we have very extreme climate gradients. So we can really look at effects of climate and the relationships of plant success as related to climate, because we can just choose areas of the landscape that might have the same species but have different climate regimes. So I think Hawaii really has a lot to offer the world in terms of us both having so many of these problems that are starting to be very problematic in other places, but also because we have the ability to really set up some really controlled experimental designs to test some of these ideas.
Thank you, Susan. Becky, would you like to add anything?
Sure. I agree with everything that Susan said. Hawaii is a fantastic model system because we do have so many environments. We can tease apart what is the effect of one factor while keeping other environmental conditions constant, and we have the low species diversity, which is unique for tropical systems. And I feel like one of the issues that we have with the invasive species, we are really the extreme. So anything that we learn here is definitely applicable to other systems. If we have successes here, I think that that's a great thing to celebrate because those are likely to be successes elsewhere as well.
Wonderful. And one last question before we wrap up. What final messages would you like to leave our audience with today? Any takeaway messages, Susan?
Well, I would just say that I think that the correlation between Becky and I's talk is coming up with these quantitative approaches, because I think just going into a system and using your best guess is often how we do things in restoration. And I think really trying to come up with tools or methods that can help us have more predictive power of success, I think will be real beneficial to the field of restoration ecology.
Yeah, I couldn't agree more with what she said. The idea of going from this gardening approach to a more strategic approach, I think has real advantages. And no matter what happens, we learn a lot from the strategic approach. We learn more about narrowing down the pathways of failure and the pathways of success, and what are the conditions under which they work best. And in Hawaii, we do have these simplified systems in the tropics. It's still within the United States boundary, so it's relatively easy to work here. But it's conditions and experiments that I think can apply elsewhere. We're not working sort of in a vacuum here. I feel like what we've learned here in these Hawaiian systems can be very applicable to other systems, because we're not talking about the case-specific examples of this species versus that species. We're talking about the general characteristics of species and the general characteristics of habitat suitability that Susan talked about.
Well, thank you, ladies, for a phenomenal webinar, and it is now time to wrap up. One final reminder that the audio and a copy of the presentation of today's session will be archived on the webinar webpage in case you would like to refer to them in the future. And now, if you could please take the short survey that will pop up on your screen, we would greatly appreciate it. This concludes today's webcast. Thank you so much for your attention.